Welcome back to Microbiology. Today we're covering Unit 8, which is going to be the characterization and classification of viruses, viroids, and prions. Now, characteristics of viruses. Well, and viroids and prions. So with all of these, we're going to have the viruses as being minuscule, acellular, and infectious agents. So acellular means that they are non-cellular, or considering in their shape. They do cause disease. They're going to lack a cell structure or structure that we normally identify as being a cell structure. They cannot metabolize, grow, reproduce, or respond to their environment on their own. And to increase numbers, they're going to have to recruit cells, metabolic chemicals, and ribosomes. So they have to actually enter into a host cell and take over that host cell, basically. Now, with viral characteristics, they're going to be, like we said, minuscule, acellular, and infectious agents. They're made up of nucleic acid cords of either DNA or RNA. They're not going to have a cytoplasmic membrane, and they're going to lack organelles and cytosol, so the protein jelly-like substance or liquid substance that's going to be found inside of the cell like cytoplasm. They do not possess the 4 processes of life, so they cannot carry out any metabolic pathways, which is referred to as metabolism. They cannot grow, so there's no growth. They cannot respond to their environment, so their responsiveness is negative, and they can't reproduce independently, so reproduction is negative. They recruit the cells that they have entered into, their host cells, metabolic pathways to increase their numbers. They can do this in 2 different states. They could do it in an extracellular state, which references a viruses outside of the cell. That's called a viron or virion. That's going to contain a protein coat or a capsid that surrounds nucleic acid. This is also known as a nucleocapsid. Sometimes viruses have phospholipid envelopes that surround the nucleoplasmid. The outermost layer is going to provide protection and recognition sites for their host cells that they're going to try to enter. The second type is going to be called an intracellular state. This references viruses inside of a cell. So they will be surrounded by a capsid that has been removed, and then the virus will exist as a nucleic acid once it enters that cell and it has its capsid removed. Usually the lysosome is going to use enzymes that almost act as acids that are going to kind of blast this capsule off of the virus. That way it's going to release that DNA, and so the DNA then floats within that host cell. So viral genetic material is going to be the primary way that scientists are going to categorize and classify viruses. So you have genetic material that may be DNA or it may be RNA, but it's never going to be both. So 2 different types of strands. You can have a double-stranded dsDNA or dsRNA. The RNA strands are going to be compatible and anti-parallel as they are in the DNA strand. Or it could be a single-stranded ssDNA or ssRNA. With those, nucleic acid genomes may exist in 2 different ways. It could exist first as a multiple linear molecule, or second as a singular circular molecule. Which type is going to be dependent upon the type of virus that you're coming in contact with. And so the genomes of various are going to be smaller than the cellular genome. Usually viruses are going to be a lot smaller. They can range anywhere from 4 to over 400 genes. So with these genomes, you can see DNA sequences. We see a viral genome here that's going to be found in this E. coli cell. Viral hosts. So only particular host cells are going to be infected by most viruses. So specificity is going to be due to viral surface proteins for proteins on that host cell. So if the host cell does not have the proteins that are going to be compatible with the virus, that virus cannot enter. Some viruses are going to be so specific that they can infect a particular kind of cell in a particular host. So it wouldn't even infect all of the cells of that host, only certain particular cells of that host. Next, viruses can infect bacteria, humans, animals, plants, fungi, and protozoa. So they have a large variety of hosts that they can infect. Viruses that infect bacteria are going to be referred to as bacteriophages or a phage. Viral size. So most cannot be seen with a light microscope. So it's really hard to see bacteria. Usually you have to be on the 100 objective, and then you multiply that by the 10 times objective of the ocular lens. That makes it 1,000 times larger. You can have hundreds of bacteria on a regular human cell. So with viruses, these are going to be much, much smaller than the bacterial cell. And so you can look at that and see approximate size in this picture. So sizes are going to range from about 10 nanometers to approximately 400 nanometers. So they are extremely small. Next, we're going to talk about the capsid morphology. Now, the capsid is going to be what covers or surrounds. It's going to be like its outer coating or its cell membrane. So it's going to be composed of a proteinaceous subunit called a capsomer. This is composed of single or multiple types of proteins. It's going to be visible with an electron microscope, and often it's going to have a triangular shape to it. So there's going to be two functions to this capsid. It's going to provide a protection for viral nucleic acid, and it's going to be a means of attachment to that host cell. As far as shapes, we're going to have three basic shapes for viruses. You can have what's called the helical shape. That's going to be where the capsomers are going to be spiral around a nucleic acid, forming kind of a tube-like structure. You could have what's called a polyhedral structure. That's roughly spherical with a shape that's going to be similar to a geodesic dome. Most common type are going to be called icosahedron, and that's about 20 sides to it. And the third type is going to be complex. That's going to contain a capsid with many different shapes. So here you can see some of these different shapes. Now, I often think when I look at these shapes, it kind of looks like almost a robotic type structure to me. You have the head of it that's going to be surrounded by that capsid, and you're going to have what's called the tail. With the tail, it's going to have those base fibers, which almost remind me of like little legs that crawl around. And then you have a base plate. Now with the viral envelope, it's going to be similar to the composition of the cell membrane. It's composed of phospholipid bilayers and proteins. These proteins are often going to play a role in host recognition, so it looks for a specific host cell or a specific type of host cell. It's going to be surrounded by a capsid. It has an enveloped virion, that's the virus with an envelope. You have a non-enveloped or a naked virion. And you're going to have what's called acquired from host cell during replication. And so it's going to come in, it's going to surround that host or this virus with the host cell's cell membrane, and it's going to hold it there until it's ready for replication and then release. So this is going to be formed from a section or a membrane from where it came in, kind of like it's going to be more of a phagocytosis where it surrounds it as it's bringing it in as a full particle. Now, classification of DNA. So we're going to have two different types of viral classification systems. So we have the hierarchical virus classification system, which is going to have its basis in viral classification on four main characteristics. Types of nucleic acid, which are DNA and RNA, presence or absence of the envelope, symmetry of the capsid, and dimensions of the variant or capsid. So higher taxa has not yet been established, like kingdom, phyla, class. They don't have like recognizable families, orders, or genus names that have been established, and specific epithets have been established. So you can have recognizable families, recognizable orders, recognizable genuses, and it has that specific epithet. So you might see, for example, things like Escherichia coli or E. coli. And so that would be its genus and specific epithet or specific species for that group. So English designations are going to be written in italics when we do this. We also have what's called the Baltimore classification system. This is going to be based on a viral genome composition and viral strategy for gene expression. So we're going to have 7 different groups. And so with this, we have that double-stranded DNA. We're going to have a single-stranded positive sense DNA. You're going to have double-stranded RNA. So looking at some of these, You're going to have single-stranded positive sense RNA, single-stranded negative sense RNA, and single-stranded positive sense RNA with DNA intermediate life cycles. And then, of course, you could have the double-stranded DNA with RNA intermediate. So positive sense is going to be equivalent to mRNA. Those are instructions that ribosomes are going to use to create protein. So they use it during translation process when creating and putting together these amino acids to make a protein. Negative sense is going to be complementary to the mRNA that cannot be directly translated or put together to make a protein. And so with these are going to be the most commonly used systems for viral classification. Viral replication is going to be hard because it has to actually be in a host cell to replicate. They cannot reproduce on their own. They do not contain genes for all enzymes that are necessary for replication is one reason, and another reason is they don't contain the functional ribosomes for protein synthesis. They're going to actually have to take over the ribosomes of that host cell. So replication is going to be dependent on the host cell's organelles and on that host cell's enzymes to produce new variants. So usually we're going to work with elliptic replication. And that usually results in the death and lysis of a host cell. You're going to have 5 main stages in elliptic replication cycles. First, you have what's known as the attachment. The virion is going to attach to the host cell, and that's dependent upon chemical attraction and precise fit between the tail fibers and of the virus and the receptors of that host cell surface. So if they don't fit perfectly, it's not going to be able to attach. Second stage is going to be entry. The virion or genome is going to enter the host cell, leaving the other parts behind. Third is synthesis. So the host cell's metabolic enzymes and ribosomes are going to be used to synthesize new nucleic acids and viral proteins of that virus. Fourth is assembly. So new variants are going to spontaneously be assembled in the host cell. This is going to involve some capsomeres surrounding replicated or transcribed nucleic acids. And fifth and last step is going to be release. So we have new variants that are going to be released from that host cell, usually due to lysing or bursting of that cell, and it releases it into that interstitial fluid or into the fluids of the body, such as blood. And from there, it's going to go into other cells and infect the other cells around. So length of time for viral replication is going to be variable. It's going to be dependent upon factors that influence metabolism with that host. So here we're looking at a basic diagram of lytic replication. And so we can see time in minutes. So once it has its entry, it goes through its synthesis, assembly, and then by the time it has enough in there to burst, we're only at 25 minutes. And so it's going to have released those variants by lysis or by bursting of that host cell here. The next type of replication is going to be lysogeny or lysogenic replication. So the infected host cell is going to grow and reproduce normally from generations before lysing. So lysogenic is going to be a temperate phages, or referred to as temperate phages. These phages are going to be involved in lysogenic replication. So we have 8 main stages to this lysogenic replication. First, the variant is going to attach to the host cell, dependent upon a chemical attraction and precise fit between the tail fibers and the virus and the receptors of the host cell surface again. So it has to match perfectly. Next, we're going to have entry. So the virion or genome is going to enter that host cell, leaving the other parts behind. The host cell's DNA is not destroyed here. The virus remains inactive or in what's called a prophage. The third step, we have the prophage in the chromosome. So it's going to be inserted into the DNA of that host cell. Fourth step, we have replication of the chromosome or virus and cellular division. So DNA is going to replicate, and so does that prophage that's been inserted into the DNA. The prophage is then going to be passed to the daughter cells once they replicate and go through mitosis. The fifth phase is going to be induction. That's where the prophage becomes excised from that chromosome, and we have what's called lytic cycle beginning. Sixth phase or sixth stage, we're going to have the synthesis. That's where the host cell's metabolic enzymes and ribosomes are going to be used to synthesize the new nucleic acids and viral proteins. The seventh phase or step is the assembly. The new virions are going to be spontaneously assembled in the host cell, and this is going to involve capsomeres surrounding the replicated and transcribed nucleic acids. And then the last step, the 8th step, is going to be release. So here we have new virions that are going to be released from the host cell, and then the host cell will lyse and die as this happens, so it bursts. Now, viruses can replicate differently or have specific steps in animals. So animal viruses are going to kind of use some of the same basic replication pathways as the bacteriophages, but they have 3 main differences between them. The first difference is the presence of an envelope that surrounds or is going to be around some viruses. Second, you have eukaryotic nature of animals. And third, they're going to lack a cell wall in animal cells. So this is going to allow for easier access into that host cell. We have 5 main stages in animal virus replication. So the first stage is going to be attachment. We have that is going to be, again, dependent upon the chemical attraction and precision and fit between the proteins and the glycoproteins on that virus and the receptors of the host cell surface. So if it doesn't fit, it doesn't enter. This is going to occur as a result of like glycoprotein spikes or other attachment molecules. So animal viruses do not contain tails or fibers in them. So if you see tails or fibers, they do not belong with the animal viruses. Second, we have entry and uncoating. So 3 main mechanisms for animal virus entry is going to be 1, direct penetration. So a naked virion is going to attach and sink into the cytoplasmic membrane. Second type, or second way, is going to be membrane fusion. That's where a viral envelope is going to fuse with the animal cell membrane, and capsid is then going to dump into the cell. And third, we have endocytosis, and that's what I was telling you before. This can involve the naked or enveloped virion, and it's going to stimulate the animal cell to engulf the entire virus. Next, we have uncoating. So once it enters, it uncoats and involves the removal of the capsid in case that the virus penetrated with the capsid intact, and it's going to be necessary before that genome of the animal virus can be activated. Now we're going into the third step. That's synthesis. Dependent upon the nucleic acid involved, if it's DNA virus, it enters the nucleus. If it's RNA viruses, it's going to be replicated in the cytoplasm. So dependent upon whether nucleic acid is a double strand or single strand, if it is a double strand DNA or dsDNA, it's similar to the normal replication of cellular DNA and translation of proteins. So each strand of the viral DNA is going to be used as a template for the complement. If it's a single strand DNA or ssDNA, it's going to be synthesized by the host cell's enzymes, which are going to synthesize the complement to the ssDNA. Complementary strands are going to bind to viral ssDNA to form a dsDNA molecule or double-stranded DNA molecule. Positive sense single-stranded RNA or ssRNA can directly be translated by the ribosomes to synthesize proteins. Complementary negative strand RNA or negative ssRNA is also going to be transcribed, and that's going to serve as a template for more ssRNAs. Retroviruses are going to contain positive SS or single-stranded RNAs. They're going to carry reverse transcriptase to transcribe DNA from their RNA. Negative sense SS single-stranded RNA is going to carry RNA-dependent RNA transcriptase. This transcribes mRNA to the negative RNA genome and enables a virus to translate proteins. And last, we have the dsRNA, the double-stranded RNA. That's where one strand of the RNA is going to be a molecule function in translation, and the other strand is going to function as a template for RNA replication. Now we're going into the fourth step. That's assembly and release. So DNA viruses are going to be assembled in the nucleus. RNA viruses are developed solely in the cytoplasm. The envelope of the animal virus is going to be released by way of budding and released through nuclear membranes, so endoplasmic reticulum or cytoplasmic membranes. The virion is going to acquire a portion of the cell membrane, which becomes an envelope around it. It's going to remain active because the host cell has not yet lysed or burst. Persistent infections are going to be like enveloped virions that are going to shed slowly and steadily within the body. You could have a naked virus, which can be released by way of exocytosis or by lysis. Now we're heading into our fifth step. Now with the fifth step, that's where the animal virus has a latency. when the animal virus remains dormant in the host cell. So it can remain there, but not active. So latent viruses, or what's called proviruses, are going to be viruses involved in latency, things such as chickenpox and herpes. They can stay in the system for years or for the rest of the life of that organism, and they can continually reactivate when that host is under stress. So it could be prolonged for years with no viral activity at all. For example, when chickenpox becomes reactivated, it's then known as shingles. So here's showing you some of the examples of the single-stranded RNA positive coming in. We have an example of the negative single-stranded RNA, and then the last one we have the double-stranded RNA that I spoke about. And here we have where it has the capsid around it, and it's going to form, and it's going to be released to the outside of the cell. And so it has that envelope, and it's going to have those viral glycoproteins that are going to be attached to it as it's released. So replication in animal viruses, in time we're going to have the entry. It's going to be assembled and synthesized, and then as it goes into its release, so it does take a little bit of time for this to occur. Next, we're going into viruses and cancer. So first we have what's called neoplasia. This is going to be an uncontrolled cellular reproduction in the multicellular animal. It can create things like tumors. We have tumors, which are going to be masses of neoplastic cells, and two different types. You can have benign, which are relatively harmless, or you can have malignant, which is going to be invasive. The malignant's also going to be referred to as cancer. It is going to be metastatic or have metastasis. That means it spreads these malignant cells to produce new tumors. There's going to be three issues that are associated with cancer and metastasis. First, it's going to rob the normal cells of space and of their nutrients. So it's going to start taking up all the nutrients, and it's going to crowd the regular cells so they can't perform their job. Second, it can cause pain. And third, it can derange the function of the affected tissue. So the body's going to no longer withstand loss of the normal function of these cells and eventually starts to die. So viruses and cancer, we have what's called proto-oncogenes. These are going to be genes that play a role in cell division. So these genes become repressed, usually, and there's no cancer. The oncogene is going to be an activated proto-oncogene. And so viruses are going to be implicated as activators of oncogenes and inhibitors of repressors. So several other factors are going to include things such as UV light, radiation, carcinogens that have the same effect on the body. Viruses cause about 20 to 25% of human cancers. One type is going to be Hodgkin's disease. Hodgkin's disease is going to be a blood cancer. It's going to occur when the lymphocytes and white blood cells begin behaving abnormally. Another type is going to be cervical cancer. That's a malignant mass that arises from cells that originate from the cervix, and this is called by the human papillomavirus, or HPV. Culturing viruses in the lab. So because viruses cannot metabolize or replicate by themselves, they can't be grown in the standard broths or agar plates. Instead, they must be cultured inside of suitable host cells. One way of culturing these viruses is in bacteria. So most viral replication knowledge has resulted from bacteriophage research. So it's easy to culture because bacteria are easily grown and maintained. These phages grown in bacteria are maintained in liquid cultures or agar plates. Plaques are going to be clear zones or areas where phages have lysed that bacteria or killed the bacteria that caused them to burst. Plaque assays are going to be a technique that enables the estimation of phage numbers. So it tells us how many phages have developed in order to kill these bacteria. Another type would be culturing viruses in plants and animals. These plant and animal viruses are going to be grown in laboratory plants and lab animals. Three issues with maintaining laboratory animals, usually they're going to be things like rats, mice, rabbits, or primates, is that it's going to be difficult, it's going to be expensive, and there's lots of ethical concerns. Should we be doing this, growing animals just intentionally to harm them to see the effects of different types of viruses and different types of cancers? Another way we can study them and culture them is by culturing viruses in embryonate chicken eggs. It's an alternative method of culturing animal and human viruses, and there's 4 reasons that chicken eggs are going to be useful to culture these as a culture medium. They're very inexpensive. They're the largest of all single cells, or a cell. They're going to be free of contaminating microbes because they're encapsulated, and they contain a nourishing yolk. So fertilized eggs are going to be the most suitable. They contain developing embryos, which contain tissues that are going to be ideal for inoculation. Next, we have another type. We can culture viruses in cells or tissue cultures. So a cell culture is going to consist of cells that are isolated from an organism and grown on the surface of a medium or a broth. There's 2 different types. You can have a diploid cell culture, and that's going to create it from an animal, plant, or human cell, so a regular cell that has all of its copies of DNA. Or you can have what's called a continuous cell culture, and that's going to be derived from tumor cells. 2 main advantages of doing this. We have them as being less expensive than maintaining animals, plants, or eggs, and here you do avoid those moral issues. So the last quandary we come about is going to be, are viruses alive? Well, some scientists believe they are, but most scientists believe they don't. They're not. So viruses are going to be considered complex pathogenic chemicals. They're also considered complex living entities. 3 observations that would suggest this is that they use sophisticated ways of invading cells. They have the ability to take control of their host cell, and then they're able to replicate themselves once they're inside of that host cell. Outside of the cell, though, they don't appear to be alive. Inside of the cell, direct synthesis and assembly required to make copies of themselves is easy. That's what they do. So viroids are going to be important in this. Viroids are going to be small circular pieces of RNA, similar to RNA viruses, but they don't contain a capsid. They're infectious and pathogenic in plants, and they have viroid-like agents that include infectious and pathogenic things that are found in fungi. So looking here, you can see the genome of a bacteriophage. That's going to be a virus that is able to infect bacteria. Looking at these small little pieces, these are actually going to be your viroids. So you can see how small they are compared to the regular genome of a virus. Now here you're going to see a regular potato on the left-hand side, and the potatoes on the right-hand side are going to be stunted as a result of being infected with this viroid infection from the PSTV that you see here. And the last thing that we're going to cover today are prions. These are going to be infectious protein particles. They lack nucleic acids, and there's going to be 2 different types of cytoplasmic membrane protein prions, or PRPs. You have cellular PRPs, which contain alpha helix, and you have prion PRPs, which contain beta pleated sheets. These beta pleated sheets are going to result from mutations of the PRP gene. They are going to be replicated by converting similar normal proteins into new prions. And so these are going to use enzymes to refold that cellular PRP within the cell. This is going to cause spongiform encephalopathies. So with these encephalopathies, you're going to involve fatal neurological degeneration, and right now we have no treatments available for these. So they not destroyed by normal cooking or sterilization if they're in like foods or meats. They are only destroyed by incineration. So if you incinerate something, then you can't intake it as a food. So there's no way to get rid of these if they do infect the food source. So this is going to be a picture of a microscope slide of spongiform encephalopathy. You see that these large vacuoles are being created. That's going to make that tissue of the brain tissue more spongy-like because it is destroying the brain tissue that is there. So different types of prion diseases. First, we have Creutzfeldt-Jacobs disease. This is going to be a rare, fatal neurodegenerative disease that's going to affect the brain. It's caused by abnormal proteins called prions that are going to accumulate in the brain that lead to progressive dementia and death. So, some of the symptoms, it starts out with memory loss, confusion. You have changes in behavior and personality. Eventually leads to difficulty in walking and coordinating movements, muscle stiffness and spasms, which then lead into seizures, hallucination, blindness, and eventually death. Next, we have Gertzmann-Straussler-Schlenker syndrome. That's a mouthful, GSS syndrome. That's going to be a rare inherited neurodegenerative disease that's going to be characterized by progressive neurological decline and then ultimately death. So it's going to be caused by a mutation in the PRNP gene, which is going to code for the prion protein PRP, like we just talked about, and make it misfold. And then it starts to accumulate in the brain, and that leads to cell damage and neurodegeneration. So some of the symptoms with this one, you're going to start out with gait and balance problems. Then you're going to lead to cognitive decline, memory loss, dementia, movement disorders, and then psychiatric symptoms and sensory loss, and then it leads to death. Fatal familial insomnia is going to be a rare inherited neurodegenerative disease that's characterized by progressive insomnia, autonomic dysfunction, and ultimately death. And so with this one, it's going to be caused by a mutation in the PRNP gene, which is going to encode for prion proteins. This mutation is then going to lead to the production of misfolding prion proteins that accumulate in the brain and then damage the nerve cells. So symptoms of this one typically begin in adulthood and progress over several years. So first they start out with insomnia, the inability to fall asleep or stay asleep. Then you have your autonomic dysfunction. So you're going to have problems with body temperature, heart rate, blood pressure, and sweating. Next is going to lead to cognitive decline. So we have memory loss, confusion, and then difficulty concentrating, motor problems, difficulty walking, you're going to have tremors and seizures, and then it could also have psychiatric symptoms like anxiety, depression, and hallucinations. And our last prion disease is going to be Kuru disease. That's going to be a rare fatal neurodegenerative disease that was once prevalent among the 4 people of Papua New Guinea. The causes of this is going to be a prion, which is going to be misfolded proteins that can accumulate in the brain and cause brain damage like the others. With these people, they traditionally practice ritual cannibalism, and that's going to lead to the spread of Kuru through the consumption of infected brain tissue. Symptoms of this one are an unsteady gait, you have tremors, loss of coordination, and difficulty swallowing. So as the disease progresses, these symptoms are going to worsen and may include things like dementia, paralysis, incontinence, and then death. Treatment for this is that there is no cure. It has a supportive treatment that's going to focus on managing the symptoms and providing comfort for the patient as they start to decline. So this is going to end us for our unit on viruses. I'll see you back with our next unit.